Hello everyone, welcome back to my channel. This is Salus Idris from MSCAN Life Science Simplified. Last days I uploaded a video on the pathogenesis of COVID-19 where I explained all the steps, all the havoc that the SARS-CoV-2 virus caused in COVID-19 infection. But actually there is something missing, and that is because this is totally new virus. So many of its activities are under discovery. So the new mechanism that is now associated with COVID-19 is hypercoagulopathy. So in today's video, I'm going to explain the coagulation process entirely. Why is that process repeatedly occurring in COVID-19? The reason for elevated D-dimer level, and lastly, why we are seeing internal bleeding in COVID-19 patients. So guys, if you would allow those things that I mentioned in their simplified form, just stay tuned to this video. I'm going to explain those things that I mentioned. Alright, let's get started. Whenever we have an injury, which may be a cut or any of that nature, what happens is that we damage some portion of our endothelial cells, and that will allow blood to move out into other spaces through those damaged regions in the endothelial cells, which we actually have to stop in order to avoid large blood loss. So these are my damaged endothelial cells after an injury is sustained. So the blood in this space will start to move out to this region which we want to block, and this can be stopped by coagulation. So let's see what happens to stop this blood flow. Immediately after an injury was sustained, a protein will be released at the point of the damaged endothelial cells, and this protein is called von Willebrand factor. What did I call this protein is von Willebrand factor. And so see how this protein will be released. So at this point, this protein will be released. So this is my von Willebrand factor protein. So in this surface, there are tons of platelets. Some are activated and some are inactivated. So these are my activated platelets. These with this green head are the activated platelets, and these red ones with no green head are the inactivated platelets. So what this von Willebrand factor protein will do is that it will stimulate the activated platelets to bind to the site of the injury. This is what I mean. So it will stimulate these activated platelets to bind to this side of the damaged endothelial cells. The binding of these activated platelets will then stimulate the release of some chemicals. Those chemicals include ADP and thromboxane A2. So this is what I mean. As these platelets, activated platelet binds to the site, it will trigger the release of ADP and thromboxane A2. So what these chemicals do is that they move into these spaces and activate these inactivated platelets. So now they will activate these inactivated platelets, and by activating them, those chemicals will also stimulate these activated platelet now to bind to the site of the injury, and by binding they will form an aggregate. This formed aggregate is called platelet plug, and this process entirely, the overall process, is called platelet plug formation. What did I call it? Is platelet plug formation. The next thing after this platelet plug is formed to prevent any further blood from going out to this damaged region is the coagulation cascade, which is the demon, right? So let's go and look into that now. At the top of this platelet plug structure, that is in the membrane of these platelets at the top, there is a compound called phosphatidylserine. So this is what I am explaining. So in this membrane of this outer layered platelets, there is phosphatidylserine. And because of this compound, phosphatidylserine, that will make the outer cover of this platelet plug structure to have a negative charge. So this structure, this platelet plug structure, will have some negative charges at the top, covered like a cap to this structure, right? So this is what I mean. So this structure will have some negative charges, and these negative charges are brought by the presence of phosphatidylserine that is found in the membrane of these platelets. So the first coagulation protein that will be moving around this surface is gluten factor 12, but it is inactive. So throughout this lecture, I will be using red color for inactivated gluten factors and green color for activated gluten factors. And also, even if I say, whether I say gluten factor or I say factor, I mean the same thing. So what factor 12 will do is that it will interact with these positive negative charges in the surface of this platelet plug structure, and it will become activated. So now we have activated factor 12. So this activated factor 12 will then convert inactive factor 11 to activated factor 11. So this activated factor 11 will then convert factor 9 from inactive factor 9 to activated factor 9. Remember, I said activated factor 9, not factor 10. Don't follow the sequence, right? So factor 11 activate factor 9. Factor 9 will then combine with factor 8 to form a complex, and this complex formed will then convert factor 10 from inactive factor 10 to activated factor 10. So now we are going further into this coagulation cascade, right? So now we have activated factor 10. So this reaction of combining factor 9 and factor 8 to form a complex that will activate factor 10 requires plasma factor 3 and calcium as cofactors. So for this reaction to occur, these cofactors are needed. What did I call these cofactors? They are plasma factor 3 and calcium. So now we'll continue with our activated factor 10, right? Factor 10 will then combine with factor 5 to form a protein called prothrombin activator. What did I call that protein? Is prothrombin activator. So this reaction for combining factor 5 and 10 to form this prothrombin activator also requires platelet factor 3 and calcium as cofactors. So now in our cascade we have prothrombin activator. This prothrombin activator will then convert factor 2 from inactive factor 2 to activated factor 2, or in other words, from prothrombin to thrombin. So factor 2 is called thrombin. Factor 2, or to say thrombin, will then react in two ways. Now I will explain the first one. In the first reaction, factor 2 convert fibrinogen to fibrin. So look at what I mean. So these red dots here are my fibrinogen molecule. Fibrinogen is a highly concentrated plasma protein. And what does factor 2 do is that it will polymerize these fibrinogen molecules into a strand protein called fibrin. So it will polymerize it into strand, and this strand is called fibrin. So the product of the first reaction of factor 2 is the formation of fibrin molecule, right? So now the second reaction. In the second reaction, factor 2 convert inactive factor 13 to activated factor 13. So what does factor 13 do is that this formed fibrin strand from the first reaction of factor 2 will be cross-linked. So this is my 3 fibrin strands. So what does factor 13 will do is that it will cross-link this strand to form something called fibrin mesh. What did I call it? Fibrin mesh. And because of this cross-linking activity of factor 13 is also called fibrin stabilizing factor. What did I call it? Fibrin stabilizing factor. So this formed fibrin mesh will then be led over to this platelet plug structure, and the essence of this is that it will hold these platelets that form this platelet plug structure so tight enough that they cannot move from these spaces, and thereby it will prevent them from causing a kind of breakage to this damage point. So if we can conclude, we can say that the function of this fibrin mesh is to hold this point of injury so tight, so make it tight enough that no blood can move out of this space, and also no platelet will move out of this space. And what happens when this damage point is sealed? That is, when this endothelial cells fully recovered and there is no any damage to it. So what happens and what we need to do here is that we have to get rid of this large structure we create, which comprise the platelet plug structure and the fibrin mesh. We have to get rid of this structure we formed, because this structure as seen actually can affect blood flow in these endothelial cells. So when this structure is broken down, it will result in the release of some small molecules. One of those small molecules are D-dimer. So D-dimer will be released. So these are my D-dimer, these three dots you are seeing, and D-dimer stands for fibrin degradation product. D-dimer means what? Fibrin degradation product. So I have one explanation here. If we have a lot of D-dimer, that means we have broken down a lot of clot, right? Because it's after we break down a clot that we will get a D-dimer, right? So if we have a lot of clot, what does that signify? That means there are a lot of injuries that are happening in that organ, right? So for this, a lot of damage to the endothelial cells will lead to a lot of D-dimer and a lot of clot occurring. So let's keep this in our mind before we head to COVID-19, caused by the SARS-CoV-2 virus. I always explain that the SARS-CoV-2 virus uses spike protein and bind to ACE2 receptor, right? And get into the lung and cause the COVID-19 infection. So now, this time is not the lung, because recent discovery shows that ACE2 receptor is also found in the endothelial cells. So what this virus will do is that it will use its spike protein and bind to this endothelial cells ACE2 receptor. How it binds to it, it will be internalized into these endothelial cells. So when it gets internalized, in the blood there are tons of neutrophils, and those neutrophils now will travel and get into these endothelial cells, and in an attempt to kill this virus, they will release reactive oxygen species, and this reactive oxygen species might end up killing some of the virus. But what they do at the end is that it will damage this endothelial lining. And if you can remember, when these endothelial cells are damaged, what we have, our coagulation cascade will start, right? Formation of platelet plug, fibrin mesh, and all those things, right? So this virus will keep attacking this endothelial lining, and as a result, it will cause a lot of injuries to it. And if you can remember, I told you if there are a lot of injuries, that means the body have to perform a lot of clothing in order to prevent large amount of blood from moving out, right? And this will consequently elevate the patient's D-dimer level. Why? Because you know that D-dimer is obtained after a damaged portion is completely recovered, and after breaking down that clot, you get a D-dimer, right? Another thing is that internal bleeding is also found in COVID-19 patients, and that is because, as you can see, there is a lot of clotting happening in those patients, and because of that, the patient might go shortage of some proteins, clotting factors, and some enzymes, right? Because of elevated use of those proteins and all those clotting factors. Because of that, COVID-19 patients can reach to a level that they can no longer perform clothing after an injury was sustained in them, and because of that, there will be excessive internal bleeding in them, that which we are now witnessing in COVID-19 patients. Alright guys, that's it for this video. I hope you found it really helpful. Please, if you have anything on this video, drop it in that comment section. I would like to see your comments and I will reply to as many as possible. And if you think others will benefit from this video, please share it with them. And don't forget to subscribe. Yeah, I always say share and subscribe. That's the only way you can support me, guys. Please do share and subscribe. And in fact, hit that bell icon so that you'll be notified anytime I make a new upload. See you next time with a brand new video. Salut, love you all. Stay safe.